Já, velkomin á Málstofu. Í dag það Dr. Steve Campana, prófessor við Háskóla Íslands sem ætlar að sýna okkur aðeins inn í leyndarheim kvarnanna. Og svo að loknu erindinu þá tími fyrir spurningar og þá berið þið þær upp í þennan mikrofón. So if you have any questions afterwards, you will use this one. So welcome, Steve. The floor is yours.
Tack för det, Lucy. Good day. Pretty much exhausted my Icelandic at this point. It's a pleasure for me to be here again, and as many of you know, I've been at the university for just 2 years now, but I recently signed on for another 4, so hopefully I will be seeing more of you over the next little while as we get going. One of my great research interests over the year has been with aging fish and with working with otoliths. And what I'd like to talk to you today is about some of the improvements that we've made for aging fish in general, but particularly some of the Sebastes species that you catch here off of Iceland as well as we caught in Canada and off of the European coast. I'm sure I don't have to tell you that the otolith is actually one of the most important structures in the fish, placed there solely to keep biologists like myself happy in their research career. They're used for many things, but primarily for aging, and they range in size from those like larval otoliths near the tip of this pin to those that are considerably larger. Now, the usual strategy when you have a large otolith is you cut it in half, you take a thin section, and you look for the growth bands. And that is widely used right around the world. And the otolith really is a primary aging structure for many species. And typically, when you go into the literature, you'll see these beautiful pictures showing these increments, these growth bands that your grandmother could age, and there's absolutely no problem. And indeed, some species are quite easy. Even the thorska here, relatively speaking, is a relatively easy species to age. But that is not the case, certainly, for other species. And the reality is that there are certain species, there are certain otoliths that are very, very difficult to interpret. Depending on what you consider to be an annual growth band, depending on which axis you look along, depending on how you prepare the otolith, if you indeed prepare it, you can get wildly different answers. So some people refer to the fish aging as more of an art form than a science, and that's not true. It's no more than an art form than, say, stock assessment modeling is an art form. There are some people that are just more suited to it, that have better aptitude for it. But certainly there's more room for rigor in terms of how these fish ages are developed. One of the groups of species is considered sort of moderately challenging around the world, and in some areas very, very challenging, are the Sebastes group. And I think your main Sebastes species here in Icelandic waters is Marinus, but I think you also have some mentella. Off of the eastern coast of Canada, we have all 3 of those species, but 2 in particular are the subject of the fishery, mentella and fasciatus. And to the naked eye, you cannot tell them apart. They are visually indistinguishable. And so typically, you either have to dissect them and to look at the musculature around the swim bladder, of all things, or you count the anal fin rays and look for statistical differences in how many fin rays are there. It's that level of detail to tell the species apart. So all of the aging studies to date have basically lumped them together. So for this study, what we did is we actually used microsatellite DNA to distinguish between all of the individuals, so we knew exactly what species we were talking about. And for the aging, the typical method that's been developed it was developed off the west coast of North America, and it's been adopted in a number of other areas, was to actually break them in half and then burn the broken surface over a flame, so-called break and burn. Crack and burn. Seems like a very ad hoc method, but it's been used for many years. And we wanted to see if we could improve on this method, or indeed decide if it was necessary, given current technology for aging fish. So what we started by doing is using a low-speed diamond-bladed saw to do a cut through the center of the otolith. we did not prepare a thin section. We just cut it in half at this point. And then we hold this otolith over the flame of an alcohol lamp and char it or burn it for about 10 or 15 seconds. And for reasons that are not completely clear, the result is something like this, where the charring seems to amplify the growth bands. Not just the annual ones, of course, any growth band, any mark on the odor. But it can give a much clearer view than, say, the unburnt vision here. And this seems to be the case for not just Sebastes, but some other species, in particular the pleuronectids, the flatfishes. One thing that we did learn when we were doing these trials with this burning is you should never, ever let a male graduate student try and use the flame. Look at the grin on this guy's face when he sees that fire. We were skeptical, I must admit, when we got into the aging. And I was quite convinced myself that using just normal thin sectioning methods with image enhancement, which I'll talk about in a bit, we should be able to match anything that the break and burn method can do. So what I've got here is some results of a matched design study that we did, where we took individual fish and we aged them in 4 different ways. We prepared the otoliths in 4 different ways, and in many cases using the same otolith. So using a high quality microscope with a high resolution digital camera, this is the view that you would get without enhancement, without burning, of one of these Sebastes otoliths. That would be difficult to age by most people. Using this image enhancement protocol that we've developed, and then we'll talk about later, there was a significant improvement of these increments. And certainly in this area here, which is the area that's very checky, is a word that the otolith readers will often use. Many, many growth marks, most of which have nothing to do with time. But down here, associated with sexual maturity, you can certainly see that there is some banding there, but it's still not very clear. When we took the same otolith and we burnt it with no image enhancement, you can see that the appearance is slightly amplified, somewhat better than what we had before. And then the final stage was to enhance the image of that burnt otolith, and you can see that things have improved dramatically. There was no question, in our matched pair design, image enhancement of the burnt otolith was significantly better than without. And that convinced the skeptics among us, primarily myself, that this approach was worth pursuing. It became quickly apparent that the image enhancement, though, was particularly important. And this protocol that we use for the image enhancement is actually something that we routinely use, not just in Sebastes, but in many different species and many different samples. And it really boils down to just 2 steps. Something known as gray level expansion or histogram expansion, and then one of the filters, unsharp mask, which was originally developed by NASA to help them interpret their deep space images. And we find routine use of these 2 steps, which takes literally seconds, can improve any image that you're dealing with. Sometimes it doesn't make much of a difference. Often it makes a big difference. And you know what? Not just for aging preparations. This combination is very effective on, say, underwater images of anything and images of the sky. Your routine snapshots that you take, you can probably improve significantly using these steps. And I won't go into great detail on these steps, but basically the first step, the gray level expansion, takes advantage of the fact that most of the image here is spread out between pure black and, say, pure white. And the typical imaging camera will divide that into 256 different levels, 256 gray levels of brightness, if you will. The better cameras will expand it into thousands. But your image, and here's the frequency histogram of the image, going from pure black over here to pure white over here, you can see there's a big peak right here associated with this background black, and the image of, in this case, the vertebral section, is compressed into a very small part of the range. So out of that 256 potential gray levels, your image information is really contained in, say, 80 or 100 gray levels. You're wasting all that space. So the gray level expansion effectively takes this image and spreads it out over the entire available space. this is not the same as increasing contrast. When you turn up the contrast on, say, your TV screen or something, you're basically doubling the gray levels. There's no new information. When you do this, you're taking advantage of the capabilities of the camera. And when it comes to the gray levels that the human eye can distinguish, about 180 or so. So 256 gray levels is more than the human eye can distinguish. When you get to the better cameras that have, say, 64,000 gray levels, you're talking about things that are physiologically impossible for the human eye to resolve. So this gives you the capability to see information that you physically cannot see without this enhancement. The second step is the unsharp mask filter. And this is basically taking your image, blurring it a bit, subtracting the original from the blurred image, the other way around, blurred image from the original, so that you see the edges and the gradients in the image, and then add that gradient back onto the original. So you're not changing anything, you're just enhancing what's there. And this is probably the most powerful filter that you can get. You know, I've shown it here from Photoshop, but ImageJ and many of the other imaging programs allow you to do this. Again, these things take seconds to run, and they are in routine use for all of our imaging. So, going back to our Sebastes, what did we find? Well, we had much clearer images, of course, but we found a very big difference. The initial structures here at the annual level are very full of false checks. Nevertheless, you can interpret them. When you get to this mature area, things were slightly different between the species. And here I've zoomed in on this mature area in Sebastes mentella, and you can see that it's fairly clear. In the fasciatus, there was two different interpretations that are possible. Let's just look at a higher magnification here. So this is the mature area of Sebastes mentella. You know, that's pretty clear cut. Each one of these is apparently an annual growth band. When you go to the Sebastes fasciatus, depending on how you interpret it, you can come up with two ways. So the pattern here seems to be towards fairly broad growth marks, but you can also interpret it that every line means an annual ring. Which one is correct? So, we took samples of these otoliths, these images, and we sent them to the best aging laboratories around the world, including those that deal with Sebastes on a regular basis. And we did not inform them of the other labs' results. We just had them provide their best age estimates. They could not agree. They could not agree between the different labs. They weren't even confident themselves. So the individual labs wrote back and said, almost without exception, we're not sure which way this should be interpreted. So we needed outside information. And luckily, we have atomic bombs. So back in the 1950s and the 1960s, the Americans and the Russians would test their nuclear weapons by exploding them on the surface or up in the atmosphere. And then they produced a suite of radioactive elements and isotopes, of which one of them was carbon, radioactive carbon, radiocarbon, or as many people often call it, bomb carbon. And it was in the form of carbon dioxide. So as a gas, it got carried in through the atmosphere, got entrained by the jet stream, and literally encircled the globe in a matter of weeks. So all living things which use carbon got exposed to this radioactive pulse, including things that form growth bands, whether it's clams or fish or trees or human teeth. They were all exposed.
Excuse me, is there some delay into the ocean?
Good question. So I'll show you this slide. So here it shows the pulse of radiocarbon in the atmosphere, peaking in, say, the early 1960s. The ocean is delayed and more muted. So in other words, the rainfall and atmospheric exchange gradually got this bomb pulse, or very quickly got this bomb pulse into the ocean, and it reached an asymptote somewhat considerably later than that in the atmosphere. And the carbon here in the ocean is constantly being recycled. So it's only going down very gradually. Radioactive carbon, by the way, has a very long half-life, thousands of years. So it's not decaying. It's just getting sequestered somewhere else. The interesting thing is, no matter where you are in the world, the exposure to that bomb pulse in the surface waters of the ocean was almost simultaneous. So here I have time frames for the Northwest Pacific and the Atlantic, the South Pacific, the Northwest Atlantic, and when you overlay them, you can see that the levels that they reach, the asymptotes are different, but the time period in which they increase is almost identical. In the ocean, it's 1958. So that period of 1958 to about 1965, that period of increase is unique in the time history. So we can go in to a growth band and look at how much radiocarbon is there. And if it's somewhere in that increasing period, we can determine the year of formation within about 1 or 2 years. It's a dated marker. It's very exact. And similarly, if you have very low levels of radiocarbon in your sample, you know it must have been produced before 1958. There's no other explanation. How do we carve out these individual growth bands? Well, we use this micromill. So the micromill is a computerized diamond-bitted drill. And it's got a stage right here, which is controlled by the computer, and it's got lateral precision in both dimensions of about 1 micrometer, 1 micron, as well as vertical, 1 micron. So you've got incredible precision, and you can carve out portions of an individual annual growth band and assay it for the radiocarbon. So the idea is then you would go to some long-lived fish, for instance, carve out core of the otolith corresponding to the first year of life, and compare it with what we know was in the environment at the time. So in this case, the yellow points are those that were carved out from 50 and 60-year-old fish and compared with what the reference chronology is saying. If our aging was incorrect, if we had thought that this fish was actually 100 years old, these points would be way offset to the side. It would be very, very clear. So in this case, our age interpretation must have been correct. And we've literally done hundreds of these bomb radiocarbon assays over the years for a whole variety of fish species, for sharks, for clams, for trees, sea pens, sea urchins, you name it, we've done it. Absolutely nothing could go wrong. So the first thing that went wrong was that it didn't match at all. So this line here is the reference chronology. That's the line for radiocarbon that we know is in the environment for the ocean. And here's the points that came out from the cores of these Sebastes otoliths. So these fish were anywhere between 20 and 70 years old, we thought, based on the counts of the growth bands. And you can clearly see that they're very offset from where the line is. We actually haven't run into this problem before. So we thought, okay, every ager in our lab is routinely told to assign a most likely age for their fish, as well as a minimum possible age and the maximum possible, to take account of that uncertainty. So we've plotted the uncertainty in those age assignments here to see if it would overlap, and those uncertainties in the interpretation cannot account for the offset for what's going on. And this had us quite baffled. And then we started playing around to see how much of a bias there is there, and it turns out it was 9 years. If you assume that all of our ages were off by 9 years, no matter if the fish was 20 years old or 100 years old, everything seems to match up fine, which is crazy. Aging doesn't work that way. So we started looking for other interpretations. And we soon realized that we were missing one crucial fact, and that is that these Sebastes weren't living in the upper 200 or 300 meters of the ocean. They were living down around 500 meters. And the bomb pulse, when it appeared, quickly got circulated in the surface layer of the ocean. But for deeper waters, it's been gradually penetrating deeper and deeper over time. And in this case, for instance, it went down between 1972 and 1990 in one profile, one location. The bomb pulse had moved down about 1,000 meters in 18 years, which is 500 meters in 9 years. 500 meters is the depth of the Sebastes. So we would expect a 9-year offset in the bomb pulse, which is exactly what we saw. So it seemed to explain our results pretty well. Our initial intent by using the bomb pulse was to make it very clear that our aging was accurate, was perfect. And because we had to do this adjustment, we're making an assumption. So we went to another age validation method, something that will be familiar to those of you that work with stock assessments. If you have a species such as cod, you've been aging for many years, if you see a strong year class show up in 1 year in the young fish, you can follow that strong year class in your catch at age matrix for many years, maybe decades. In the case of the Sebastes off the east coast of Canada, there was a very strong year class in the early 1980s that was apparent in the very young Sebastes. And the stock assessment people were still seeing it 21 years later in their catch at age matrix. So those are the fish that when we aged them, we came up exactly that year class. So we know based on this that our aging up to 20 years was correct, and that overlapped with the range for the bomb dating as well. So we inferred then that our aging is correct up to about 60 years for Sebastes. I'm not going to get into the results at all. Basically, though, the two species had very similar growth trajectories. They shot up fairly quickly, and then they leveled right out. They basically almost stopped growing after sexual maturity. And Sebastes mentella grew somewhat larger, and it grew to older ages, but otherwise, they were pretty similar. So what's the bottom line? Well, first of all, much to our surprise, the combination of the break and burn with image enhancement provided the best possible way of interpreting the age of these Sebastes. We're not suggesting, and it's not true, that that combination is going to apply to other species. If you try and use a burn method on cod, it'll look a little different, but it doesn't help. As a matter of fact, in some cases, it makes it worse. So it seems to be species specific. And there are certain groups of species, like Sebastes, like the flatfishes, and some other species where it really helps. But you know what? We can't tell in advance if it's going to help. It's a very strange situation. We don't know why it's helping in some species and not in others. The image enhancement, though, is certainly very important to that. Secondly, I think it underlined what many in the otolith world and the age reading world already knew. There are certain species that are just tough to age, and there's some that are almost impossible. Lophius, monkfish, is one of those, for instance. And even the most experienced of the fish agers have trouble with them. You're not going to get around that. I'm a big proponent of the fact that you cannot trust models, ecological models, stock assessment models, without some way to calibrate against reality. And it's the same thing with the fish aging. Fish agers, when they've been working for a long time, they get very certain in their interpretations. And I'm certainly the same way. And they become very precise. If you give them a set of otoliths that they've aged a few years previous and ask them to age again, they'll probably become very close on average to what they had the original time. The reality is, though, that in various places around the world, they become very precisely wrong. They have no way to calibrate to find out if their age interpretations were correct, and therefore they become very focused on their interpretation. The age validation is the only way to find out this outside information. And this is not a reflection on the age reader. And don't get me wrong, most of these experienced age readers are going to be on the correct answer. But until you have that outside information, how are we supposed to know? It's like an ecological model. Unless you can always get results out of the model. How do you find out if it's correct? And then, not surprisingly again, Sebastes, a deep sea species. It is slow growing, long lived. So, we watch with alarm the movement of many fisheries into seamounts and deeper water areas, continental slopes, where the fishermen are encountering these large virgin biomasses and thinking, oh, wow, we can go crazy there. We've got a plentiful stock here. But in actual fact, if they're very long-lived and slow-growing, like orange roughy, once the initial fishing has removed that virgin biomass, the recruitment can take decades or centuries to replenish it. So I think we really have to watch these deep water fisheries closely because they're so unproductive. Thank you very much.
A very interesting talk. We have some time for questions. Clara?
Hi, thank you very much, Steve. Maybe a very stupid question, but I have to ask. I'm sorry, I'm Clara from Hafró. Is there any other natural pulses that we can test, like eruptions? Because this is now a little getting too late for us to use. And do you know if there is any other pulses that we can measure in same fashion? Thank you.
Not a stupid question at all. That's an excellent question and one that we raised a number of times, and certainly when people such as myself and my colleagues, we saw Chernobyl go crazy and then Fukushima a few years ago, we rubbed our hands with glee and thought, oh, that's a mutated bark. But you know what? It's not spewing out the right isotopes to pick up. So for instance, those were contained, they didn't release any gaseous cloud. And the radioactive isotopes they released are mostly those of cesium and lead. And unfortunately, they're just not present at very high levels in the fish oil. So in principle, you would think they would be there, those markers, but they're not so far. Now when it comes to volcanic eruptions, possibly, that's one of the things that we would look at to see if maybe there's some isotope or element, rare earth element perhaps, that gets released from a volcanic explosion that would be taken up by the local fish. That's a possibility. But not every element that's in the water gets absorbed into the otolith sort of in a proportional fashion. It's only a handful that do that. So we're on the search for these markers, and people are aware of it. So far, nothing. I think maybe Donald Trump, when he...
This image enhancement, is it being used routinely on labs around the world?
To the point where some labs have actually set up their image analysis systems for their agers, because all the agers age through the system, not through the microscope. They've got those enhancement protocols hardwired in, so you get a real-time image on the screen of the image enhanced view, and they age directly from that. Yeah, it's in routine use. The number of labs now that age directly through the microscope without using imaging of some kind is going way, way down.
This is Anna Ólafsdóttir at Hafrannsóknastofnun. Thank you, Steve, for a very interesting talk, and I'm glad to hear that you're staying here for 4 more years. That's great for the science community. And I have a question related to Clara's. Talking about nuclear bombs, like there was a big bomb in '45. Why didn't we see the signal from those 2 bombs in the graph? Because it went back to 40.
Okay, another good question. In actual fact, you do, if you look in enough detail. So the problem is that for the chronologies that I presented there for bomb radiocarbon, it's just an annual mean. And the introduction of that first bomb, those first 2 bombs in 1945, were relatively small in the big scale of things. When they started doing the later tests, the radioactive output was orders of magnitude larger. So if you actually look at the bivalves around in the late 1940s, you could see the initial little blips from those 2 bombs. And then as the testing picked up, you start to pick them up. And there are certain sequences, certain years, where if you look in detail within a year, you can see individual bomb events taking place. So they're there, but you have to look in, with the detail to do it. And certainly when we do the fish otolith work, we're not trying to pick out anything at a sub-annual level. We just want the individual year. And there, that contribution for those first few bombs is just too small.
Thank you.
Ella Tavro, I have a question about, you mentioned like flatfish otoliths. Do you think you can burn like the juveniles after you have polished them and then enhance the pictures?
You could, and we have done so. We've done sort of large-scale aging studies on, say, Atlantic halibut and some of the other fluoromectas. And just to be consistent, we will burn all of the size groups, but it doesn't help with the juveniles. You know, usually there, it's very clear without it, the burning risks, you know, doing some breakage, and it brings out a bunch of the growth bands you actually don't want to count. So really the advantage of the burning comes when you get to the older age fish. That's really where it kicks in.
Hi, I'm Jeremy Colley, fellow Canadian,
Hi, Jeremy.
visiting here. Nice to see you again. I wondered, with the break and burn method, is there any difference in the chemical composition of otoliths that might explain why they respond differently to the burning method?
Yeah, good question. Otoliths, in general, are a very pure structure. Matter of fact, when first people started looking at the chemical composition, the geologists were thinking, oh, this would be a wonderful standard for my electron microprobe or something. It's so pure calcium carbonate. So the trace levels there are very, very small, just a few percentage points. I think what's happening with the burning is something to do with the organic matrix. So the best understanding that we have, it's not the trace impurities that are causing the change. It is something about the ratio between organic and inorganic in the otolith. And that's what the charring is changing. And again, people still don't have a mechanism in terms of what it is that's burning, but it makes sense that it wouldn't be the calcium carbonate. That's a rock. You can't burn the rock. So it's got to be something organic in there. And there's something different about certain taxa of fish. And we just don't know. So in a big sense, yes, you're right. It's got to be something to do with composition. But it may not be the actual specific composition. It's just the ratio organic to inorganic.
Thanks.
Georg here from the Marine Research Institute as well. You showed two pictures of two possible interpretations of counting the rings on the otoliths earlier, which you then used the C14 method to verify. Which one was the actual way that you counted in the end?
It changed. It varied. It was a little disturbing. It was not consistent. There were some fish where it turned out that counting them all was the appropriate method, and I'd say most of them, that was the case. In others, it was sort of the broader pattern that you had to follow, and it basically depended on how old the fish was once you saw that pattern. So for many fish agers, they'll rely on the broader pattern. It'll usually be the correct age, and that was the case for Sebastes up to, I think, about 30 or 40 years. After that, they were so fine that basically you had to count everything. And so at that point, it became very clear-cut. You had to count all of the growth bands. And again, younger fish, you would never do that. You would come up with wildly inaccurate ages. But if it was really slow-growing fish, that turned out to be it.
So is this how you age the sharks?
That's right, I get up close and personal. We do a lot of shark aging, though, actually, with the vertebrae. And the vertebrae of some of these shark species are actually more clear than the otoliths of some fish species. So they can be very clear-cut. The challenge with the shark vertebrae is they're not otoliths, they are metabolic. So we're finding with some old sharks, they stop growing altogether. So they have an apparently clear growth sequence, and you think, oh, this is wonderful, it's 21 years old. And then when we do the bomb dating, or if we get a tag recapture after 30 years, which has happened, you realize you've missed half of the increments. And they're not narrow, they're just not there. They stop forming altogether. The sharks are a special challenge, and that's why we have to get in there.
No, it was interesting to hear about the Greenland shark. There were some results last year of old decade,
Yeah.
hundreds of years.
Take it with a grain of salt.
Okay.
I was actually, I guess I could admit this now, I was a reviewer for Science for that paper, along with one of my former students. And we weren't going to take it because there's certain assumptions that go into that paper that really can blow everything out of the water. Nevertheless, there's really nothing else on how to age Greenland sharks, and to make the point that they're really old seems reasonable. But I and the others do not accept this 400-year longevity. 100 years, maybe. So remains to be seen how to do it. And I do have an idea how to age these, and I'll tell you once I get into science.
Okay. Well, thank you, Steve, very much. And we're happy to have you as a neighbor for the next years, as Anna said. And hope to see you soon. And thank you all for coming. Thank you.